Carbohydrate metabolism, which is lecture 2 in session 2 of metabolism module. Learning objectives of this lecture is general structures and functions of carbohydrate, how dietary carbohydrates are digested and absorbed, why cellulose is not digested in the human gastrointestinal tract, the glucose dependency of some tissue, the key features of glycolysis, why lactic acid lactate production is important in anaerobic glycolysis, and how the blood concentration of lactate is controlled. Carbohydrate is a large class of compounds that are polyhydroxyaldehyde, polyhydroxyketone, or substances that yield such compounds on acid hydrolysis. The empirical formula of the carbohydrate is CnH2O by N, and the best example here is glucose, which contains 6 carbon atoms, 12 hydrogen atoms, and 6 oxygen atoms. The functional group of carbohydrate are the carbonyl group of a carbohydrate is either aldehyde, which is C double bond OH, or ketone, C double bond O. The other functional group is alcohol hydroxy group. These are the alcohol hydroxy groups. Carbohydrate can be classified into sugars and non-sugars, and the difference between them that sugars has a sweet taste, while non-sugars has not sweet taste. Sugars also can be classified into monosaccharide and oligosaccharide. Monosaccharide classified according to the carbonyl group that has, for example, aldoses and ketoses. Oligosaccharide can be classified into disaccharide, and tetrasaccharide, while polysaccharide, non-sugars, could be heteropolysaccharide or homopolysaccharide. Carbohydrate is important for the living organisms, and that is because polysaccharides serve for the storage of energy, for example, starch in the plants and glycogen in human, in addition to that structural components like cellulose in the plant and chitin in arthropods. 5-carbon monoxide can enter in the synthesis of the coenzyme, for example, ATP adenosine triphosphate, FAD flavin adenine dinucleotide, and NAD nicotinamide adenine dinucleotide. In addition, 5-carbon monosaccharide ribose considered as the backbone of the genetic molecules RNA and DNA. Saccharides and their derivatives play a key role in the immune system, fertilization, blood clotting, and development. This slide showing the difference between the 3 types of the carbohydrate, monosaccharide, disaccharide, polysaccharide. The difference is in the subunits. Monosaccharide 1, disaccharide 2 subunits, while polysaccharide containing many subunits. The main function of the monosaccharide is for energy source, disaccharide for transport, and polysaccharide mainly for storage. And these are the examples: monosaccharide glucose, galactose, fructose; disaccharide lactose, saccharose, and maltose, while polysaccharide like cellulose, glycogen, and starch. Monosaccharide is the simple form of the sugar of carbohydrate, and these can contain from 3 to 9 carbon atoms. They are either aldoses, for example glyceraldehyde, which means containing aldehyde group, or ketoses like dihydroxyacetone, which containing a ketone group. Since monosaccharide can exist in several different forms, so it is very important to provide the complete name of these monosaccharides. So for the example here, glyceraldehyde containing 3 carbon atoms, so it is a triose, and because of the presence of the aldehyde carbonyl group, so it is an aldotriose. The other example is glucose containing 6 carbon atoms, so it is a hexose, and because there is an aldehyde group, so it is aldohexose. The other example is fructose fructose containing 6 carbon atoms, and because of the carbonyl group is a ketone, so it is a ketohexose, ketohexose. The isomerism of monosaccharides either D or L isomerism, and this depends on the orientation of the hydroxyl group that attach to the carbon atoms before the last one. So if the hydroxyl group present on the right side, so it is D-glucose. If it is oriented on the left side, so it is L-glucose. Almost all sugars occurring in a human are D-glucose. Glucose has 2 epimers, mannose and galactose. They are similar to glucose in their structure, the difference only in the orientation of the hydroxyl group in the mannose, which attach to the carbon atom number 2. This hydroxyl group oriented on the right side, while in the glucose, the hydroxyl group that attached to the carbon atom number 2 is oriented to the left. While in galactose, the hydroxyl group that attached to the carbon atom number 4 is oriented to the left. In glucose, it's oriented to the right. The other form of the monosaccharide is the cyclic form, which is either alpha or beta. And this cyclic form is due to the reaction between the carbon atom number 1 with the carbon atom number 4 or number 5. And also we have here alpha glucose depending on the orientation of the hydroxyl group that attached to the carbon atom number 1. If it is not on the same level of the hydroxyl group of the carbon atom number 5, so it is alpha. If it is on the same level, so this cyclic structure is beta. للسهولة احنا نقول إذا كانت الهيدروكسيل جروب أسفل الحلقة فإذا هي ألفا جلوكوز. إذا كانت الهيدروكسيل جروب في أعلى الحلقة فإذا هي بيتا جلوكوز. This slide showing that the types of monosaccharide which containing aldehyde group and those which containing a ketone groups. So for example here, glyceraldehyde, ribose, glucose and galactose. All these are monosaccharide containing the carbonyl group is aldehyde. While dihydroxyacetone, ribulose and fructose, these are monosaccharide containing a ketone group. Disaccharide is a carbohydrate containing 2 subunits of monosaccharide. So for example here the saccharose is a disaccharide containing glucose and fructose. Lactose, disaccharide containing galactose and glucose, while maltose is a disaccharide containing 2 subunits of glucose. Regarding polysaccharides, could be cellulose, starch, and starch classified into amylose and amylopectin, or another type of polysaccharide is the glycogen. They are different in the source, in the subunit, and in the bonds, and in the presence or absence of the branches. So, the source of the cellulose is plant, and the starch plant also, for both amylose and amylopectin. Glycogen is animal. The subunit of the cellulose is the beta-glucose, while the remaining starch and glycogen are alpha-glucose. Regarding the bonds, in the cellulose and the amylose, the bond is 1,4, which means that carbon atom number 1 attached to the carbon atom number 4. While in the amylopectin and glycogen, there is a carbon atom, there is a bonds between carbon atom number 1 and carbon atom number 4. This is in the main chain, and carbon atom number 1 with carbon atom number 6 in the branch chain. So therefore, there are no branches in the cellulose and amylose, but there are branches in the amylopectin and glycogen. In amylopectin, per 20 subunits and glycogen per 10 subunits, which means that every 10 subunits the branch will start. This is the diagram of the cellulose amylose, which is a straight chain, while in the mylopectin and glycogen there is a branch chain. What is the main sugar in the blood? The major sugar found in the blood is glucose, but fructose and galactose may appear for short periods in the blood, and that depending on the dietary intake of fruit and dairy products. Excessive amount of galactose and fructose in the blood lead to certain clinical problems like galactosemia and fructose intolerance, and these two conditions we'll talk about it later. And also glucose if present in the high level in the blood. This condition is seen in untreated cases of diabetes. The stage 1 of carbohydrate metabolism is started from the digestion of the carbohydrate. And as you know that the dietary carbohydrate could be monosaccharide, disaccharide, and polysaccharide. Monosaccharide is absorbed directly from the small intestine. Disaccharide digested in the small intestine by the certain enzymes, lactase for the lactose, saccharase for the saccharose, maltase for the maltose. While polysaccharide digested first in the oral cavity by the salivary alpha amylase, forming dextrins, and this dextrin it will be digested by the pancreatic alpha amylase into maltose. Then maltose digested by the enzyme maltase to form 2 units of glucose. All carbohydrates are absorbed in the form of monosaccharide. So these monosaccharides are 80% glucose and 20% fructose and galactose. The absorption of glucose and galactose is by sodium co-transport mechanism, while fructose is transported or absorbed by the facilitated diffusion. So these monosaccharides absorbed leaves the epithelial cells by facilitated diffusion and enter to the bloodstream. So when disaccharide present in the lumen of the small intestine, for example here the maltose will be digested by the certain enzymes known as maltase, give 2 units of glucose. Glucose, it will be absorbed into the intestinal epithelium by the active transport with sodium, while fructose is transported into the intestinal epithelium by the facilitated diffusion. And in facilitated diffusion, we're using certain types of transporter, which is called glucose transporter 5. Then glucose, fructose, and galactose now present in the intestinal epithelium. These monosaccharides, it will be absorbed to the blood circulation by the facilitated diffusion using this active transport, which is glucose transport 2, that facilitate the diffusion of the fructose, glucose, galactose to the blood circulation to be distributed throughout the body. So this figure reveals the absorption mechanism of glucose, galactose, and fructose. So as you see here that these are the keys, these that sodium glucose transport, and the light blue, the dark blue is glucose transport 2, and the purple color circle is the glucose transport 5. So you see here that the glucose or galactose, it will be enter into the epithelial or intestinal mucosa by the active transport with the sodium. The mechanism of this is using the sodium potassium ATPase. So when sodium enter when the potassium enter into the lumen or intestinal mucosa sodium it will be come out. This mechanism or this process occurred by the action of the AT-base enzyme. Therefore, the sodium concentration inside the intestinal mucosa will drop and become less than its concentration in the intestinal lumen. And because of this, the sodium in the intestinal lumen will be entered to the intestinal mucosa. With the entrance of the sodium to the intestinal mucosa, one molecule of glucose or galactose enter with it. Regarding the fructose, the absorption of the fructose from the lumen of the intestine to intestinal mucosa is by the facilitated diffusion. And this in facilitated diffusion using a certain receptors or transporter, this transporter named glucose transporter 5, which facilitate the diffusion of fructose from the lumen of the intestine to the intestinal mucosa. Now, in the intestinal mucosa, both monosaccharides are present and absorbed to the circulation by the facilitated diffusion using the glucose transport 2. So, transport from the absorptive cells into the blood and from the blood into the tissue is by facilitated diffusion, and this involves family of glucose transport protein from glucose transport 1 to glucose transport 5. The synthesis of some transporter is enhanced by the insulin, for example, glucose transporter 4, that present in muscles and adipose tissue. This figure showing that the different types of the glucose transport is distributed in different types of tissue. So for example here, in the brain there is a glucose transport 3, while in the fatty tissue and skeletal muscles, heart, glucose transport 4, that facilitate the diffusion of the glucose, while in kidney and jejunum there is a glucose transport 5, erythrocyte GLUT1, and in the intestine, pancreatic beta cells, liver, GLUT2. Cellulose is not digested in the human of gastrointestinal tract. Why? And this is because the human gut does not have the cellulase enzyme. This cellulase enzyme is responsible for the digestion of the cellulose. So therefore, the cellulose provides fiber which is necessary for the process of defecation. In contrast, cellulose can be digested in the herbivores, for example cows and horses, because they have the cellulase enzyme that can break down the beta-1,4 glucosidic bond. The minimum amount of glucose required by a healthy adult on a normal diet is about 180 grams per day. 40 grams for the RBC, YPC, kidney medulla, and lens of the eye, because they are only use glucose for energy production. 140 grams for the nervous system, because it prefer glucose for energy production, and variable amounts are required by some tissues of the, for example, adipose tissue, to synthesize glycerol. The second part of carbohydrate metabolism, or the second stage of carbohydrate metabolism, is the glycolysis. Glycolysis is a series of reactions that extracts energy from glucose by splitting it into 3 carbon molecules called pyruvates. And the name glycolysis, glyco from sugar and lysis from break. Glycolysis takes place in the cytosol of the cell, and glycolysis is the only source of energy or of ATP in the cornea and the lens of the eye, renal medulla, and RBCs. So the glucose, once the glucose enters into the cell, in the cytosol, the glycolysis process takes place. Glycolysis, it will break down the glucose into two 3 carbon molecules called pyruvate. Also, glycolysis is one of the main processes that involved in the cellular respirations. And what I mean by cellular respiration is a set of metabolic reactions to yield ATP in the mitochondria. So therefore, cellular respiration, including 4 stages, which are glycolysis, pyruvate oxidation, citric acid cycle, and the last one is oxidative phosphorylation. There are 2 main phases in glycolysis. The first one is energy investment phase, and the second one is the energy releasing phase. In phase 1, glucose, it will be reacted with the hexokinase to form glucose 6-phosphate. In this reaction, we use 1 molecule of ATP. Then glucose 6-phosphate converted into fructose 6-phosphate by phosphohexose isomerase, and then fructose 6-phosphate by the action of the phosphofructokinase 1 become fructose 1,6-bisphosphate. Also in this reaction, we use 1 molecule of ATP. Then fructose 1,6-bisphosphate by the action of the enzyme aldolase give glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Both of these molecules containing 3 carbon atoms. Then dihydroxyacetone phosphate converted into glyceraldehyde 3-phosphate by triose phosphate isomerase. So therefore, in this phase, we consumed 2 molecules of ATP. In phase 2 of glycolysis, glyceraldehyde 3-phosphate converted into 1,3-bisphosphoglycerate by the action of the enzyme glyceraldehyde 3-phosphate dehydrogenase. Then 1,3-bisphosphoglycerate converted into 3-phosphoglycerate by phosphoglycerate kinase, and here there will be release of ATP in this reaction. 3-phosphoglycerate by the action of the phosphoglycerate mutase become 2-phosphoglycerate, and this by the action of the enzyme enolase forming phosphophenol pyruvate. Phosphophenol pyruvate by the action of the pyruvate kinase forming a pyruvate compound. Also in this reaction, there will be release of 1 molecule of ATP. So, because in the first phase we gain 2 molecules of glyceraldehyde 3-phosphate, so therefore in this phase we gain 2 molecules of the pyruvate and 4 molecules of ATP. So the total outcome of ATP from this glycolysis is 2, and the cause for that because we consumed 2 ATP in the first stage, and there is 4 ATP released from the second stage. So the difference between them will be 2 ATP. So the end product of glycolysis is the pyruvate. Therefore, there are 3 phases of pyruvate that produced by the glycolysis. The first one is aerobic oxidation, which means that the pyruvate forming an intermediate compound which is acetyl-CoA, and this acetyl-CoA enters into the citric acid cycle to produce energy. and this happened when there is sufficient amount of oxygen. Or pyruvate could be enter anaerobic circulation, and this is forming by the lactic acid fermentation, forming lactate, and I mean by here anaerobic, there is low level of the oxygen. Or could be by alcoholic fermentation, the pyruvate forming ethanol. Also this happened or this occurred in anaerobic conditions. Regarding the regulation of the glycolysis, there are 3 enzymes responsible for the regulation of the glycolysis pathway. The first enzyme is the hexokinase. Hexokinase is responsible for the reaction between conversion of the glucose into glucose 6-phosphate, and in this reaction this reaction consumed 1 molecule of ATP. Therefore, the activator of this enzyme is the AMP-ATP, while the inhibitor is the glucose 6-phosphate. And this means that when there is a high amount of glucose 6-phosphate, this will stop the activity of this enzyme, and then it will stop the reaction of conversion of glucose to glucose 6-phosphate. The other regulatory enzyme is phosphofructokinase. Phosphofructokinase is used for the conversion of the fructose 6-phosphate into fructose 1,6-bisphosphate. Also, this reaction needs 1 molecule of ATP, or use or consumed 1 molecule of ATP. Therefore, the activator of this enzyme is AMP-ATP and insulin, and this means that when the level amount of the ATP is low, this will activate the enzyme to react fructose or to convert fructose 6-phosphate to more fructose 1,6-bisphosphate, while the inhibitory effect of this enzyme is ATP, citrate, and glucagon hormone. The other enzyme, regulatory enzyme, is pyruvate kinase, and this pyruvate kinase is used for the conversion of the phosphoenol pyruvate into pyruvate. And here there is a releasing of ATP, so therefore the activator is the AMP-ATP, and the inhibitor is the ATP and acetyl-CoA. This slide showing the aerobic respiration diagram. So as I said before, that glycolysis, which happened or occurred in the cytosol of the cells, is in order to form pyruvate from glucose, and the amount of the pyruvate formed here from the glycolysis is 2 molecules, in addition to 2 molecules of ATP and NADH, 2 molecules of NADH, which is an electrons carrier. Then pyruvate in aerobic conditions enter into mitochondria. In the mitochondria, it will be converted into acetyl-CoA plus 2NADH. Then this acetyl-CoA enters into the citric acid cycle, and citric acid cycle gives us 2 ATP plus 6 NADH and 2 FADH. Then these electron carriers, 2 NADH from the glycolysis in the cytosol plus 2 NADH from the mitochondria and 6 NADH and 2 FADH that result from the citric acid cycle, it will be entered into the electron transporter chain by the oxidative phosphorylation, forming 34 molecules of ATP. So the total amount of the ATP that gained from the aerobic respiration is 38. Therefore, total energy yield of aerobic glycolysis, from the glycolysis we gain directly 2 molecules of ATP and 2 NADH, and these 2 NADH give 6 ATP. From pyruvate oxidation, 2 NADH and also these two NADH give 6 ATP. From citric acid cycle, directly 2 ATP and 6 NADH, which give 18 ATP. 2FADH2 give 4 ATP, so the total will be 38 ATP. All NADH and FADH2 converted to ATP during electron transport chain by the oxidative phosphorylation stage of the cellular respiration. And each molecule of NADH converts to 3 ATP, while each molecule of FADH2 converts to 2 ATP. And this is because FADH enters the electron transport chain at lower levels than NADH. So what is the difference between aerobic respiration and anaerobic respiration? In anaerobic respiration, the reactant is glucose, while in the aerobic, it's glucose and oxygen. The combustion in anaerobic is incomplete, while in aerobic is complete. The energy yields low 2 ATP in anaerobic, while it will be high 36 to 38 ATP in aerobic. The products in anaerobic in animals produce lactic acid and yeast ethanol plus CO2, while the aerobic there is CO2 and H2O in addition to ATP. The location of the anaerobic in the cytoplasm, while the location of the aerobic in the cytoplasm and the mitochondria. The stages for the anaerobic is glycolysis and fermentation, and here fermentation either lactic acid fermentation forming lactic acid or alcohol fermentation forming ethanol, while the aerobic stages are glycolysis, link reaction, Krebs cycle, which is citric acid cycle, electron transport chain, which is oxidative phosphorylation, the other name. What's the importance of lactate production? Lactate production is very essential, it's very important to continue glycolysis when there is oxygen in low level or insufficient amount of oxygen, and this is occur during contraction of the muscle. So here about 50 grams per day of lactate are produced, and these are produced from the RBCs, muscle, under anaerobic conditions, and this lactate will go to the liver for synthesis of glucose by another pathway known as gluconeogenesis, or go to the heart muscles to be oxidized and forming pyruvate and then CO2. And then this pyruvate enter into the gluconeogenesis pathway in order to synthesis or to form glucose. So this figure showing the pathway of lactate formation. So here in the muscles, during exercise or during muscle contraction, the glucose, when there is insufficient amount of glucose, insufficient amount of oxygen, the glucose will be entered into lactic fermentation and forming lactate. This lactate, it will be absorbed to the circulation and then enter into the liver. The lactate in the liver converted into pyruvate, or it will be enter another pathway called gluconeogenesis pathway in order to produce or to form glucose, and then this glucose return back to the circulation to be used again by the muscles. So there are certain situations that lead to an increase in the plasma lactate. These are due to an increased milk production or decreased utilization of the lactate. So the causes that lead to an increase in the production of lactate include strenuous exercise, sepsis, shock, and congestive heart disease, while those that lead to decreased utilization of lactate, liver disease, thiamine deficiency, alcohol metabolism. Thank you very much for listening.